thought that you had to meet 3 criteria to be invited to do this sort of a talk or to write a prefatory chapter. And the 3 criteria were first, that you were completely done with research. Second, that you weren't dead yet. And third, that you weren't brain dead. Now, I can't judge really the first and the third criterion, but I can quote Monty Python. His line on the second one is, "We're not dead yet." Okay, anyhow, it's supposed to be somewhat autobiographical. We have to start someplace. So we'll start with Minnesota many years ago, and my own fascination with wildflowers. That was because of my father, who was he was a schoolmaster, but he was a passionate amateur botanist. And he was trying to take pictures of all of the wildflowers that he could in Minnesota. And we had discovered a piece of swampland, a huge piece of swampland, not too far from Savage, Minnesota, along the Minnesota River. Very, very rich soil. And there I learned that there were some spectacular orchids that grew. Minnesota has a wonderful collection of them. And so, for example, this guy, and among other things, my father learned how to make color prints, which was something that in those days took 2 days per print. It was a dye transfer process that was appallingly complicated. But anyhow, he taught me how to do this. I don't show anyone because they were all terrible. He taught me how to do this, and I learned a whole lot of chemistry in the process. So this is a very nice little white lady slipper. This Cypripedi's mandarin. This is a showy lady slipper. It's Minnesota state flower. And this is a very rare Habenaria, very fringed orchis. It's on the endangered species list. And here's another one that's very rare, the so-called ramshead lady slipper. Well, so I got fascinated by all of this, and this interested me in going on to become a taxonomist. Well, so we have to look at something besides orchids as a wild rose. Anyhow, to continue, we became very friendly with Ernst Abbey, who was a botany professor at the University of Minnesota. And he had a very gruff manner, but he was an absolutely wonderful, supportive guy. And I went off to college, and I was going to go to Harvard. Well, I should mention, I went to Harvard I told my father that I wanted to go not to Harvard, to Princeton. And he said, "That's okay, you can go to Princeton, but if you go to Harvard, I'll pay for it." So anyhow, every time we met Abbey, we met him lots of times. He actually took us to Itasca State Park to see that ramshead lady slipper, and step across the start of the Mississippi River, which you can do there. Every time I saw him, he'd say, "Have you taken a botany course yet?" And I was taking all kinds of other things. I was taking science, but I hadn't taken any botany courses: chemistry, physics, math, calculus. So then, in 1948, why haven't you taken a botany course yet? So finally, in the fall of 1949, I had a response for him. I took it essentially just to make him shut up. And that was, well, anyhow, I got hooked, not surprisingly. So I started out, and my family was musical. My mother was a professional. Her last professional concert, she was 83, I think. My sister was a professional. She's at 82. She's still performing and teaching. And so I thought I'd be a music major too. She'd gone through the music program at Harvard. And, well, it didn't work so well. And I wanted to be a pianist. Finally, I had to go out of denial and say, well, I really wasn't very good. And I wasn't going to play Beethoven concerti with great symphony orchestras. In fact, I wasn't even going to play with trivial symphony orchestras. Now, this is something that actually people in the music department had hinted at. It shouldn't have been complete news. But anyhow, it turned out that I could complete a music major without that, and also complete a major in biology. So that's what I did. You couldn't do that anymore because there are so many requirements you have to meet. So now we have a biology major at Harvard, and then graduate work. Now I intended to be a taxonomist, and I had from the very start. But I did get sidetracked into certain things like joining the Harvard Mountaineering Club. This is on a fantastic cliff in New Hampshire called White Horse, across from a lake. It's called Echo Lake. get on the other side, it has a wonderful echo. And, oops, wrong way. That's me, believe it or not. Along with the tango, but we've just finished the climb. And those are some of our fellow Harvard Mountaineering Club members. Well, the next thing I knew, it was 1952, and we were headed for Alaska. And this is the McKinley Range. There's Mount McKinley. The highest peak is the south peak there. Mount Forker, 17,000, McKinley over 20,000. But our objective was actually to go over to the, further to the east and to try to climb Mather, Deception, or Brooks. Now my excuse for doing this and taking an entire summer off to do it was that I was going to collect plants and find an area that hadn't been really carefully explored. And so I would be the expedition botanist. That, of course, was a bunch of nonsense. But anyhow, I did collect plants. That's me. There's a little saxifrage. And in fact, some very nice things there. This is a little poppy growing. This is the highest vascular plant we saw. I think it was in 8,000 feet or something like that. A beautiful little thing. Then you had this little saxifrage and another saxifrage and a little sunny campion. And that, of course, is what I did, but not enthusiastically. This is what I really wanted to do, and so we did it. This is now climbing Mather. And you get up on top. We already climbed Brooks over here. We're over 12,000 feet now. And there's McKinley off in the distance with the Alpine glow showing 11 o'clock at night. And that's the sort of thing one could see only from up there. Fortunately, we're on the top of Mather here, because I don't think that any of us would have cared to do this to get over to that peak. So fortunately, we didn't have to. There were hazards. When we climbed up Brooks, I managed to slide 500 feet on my tail. But fortunately, was stopped by somebody else with an ice axe. And this is actually a 40-degree flare ice slope, going across on crampons. But anyhow, we survived. And guess what? I got a paper out of it. Now, to my knowledge, that paper had never been cited. But then the next thing that happened is that I decided that I didn't really want to spend my life in a herbarium. And I took a course in plant development from Ralph Wetmore, who was working on what we now refer to as plant stem cells, 2 apices. And I was absolutely fascinated, and I got tangled up with a brilliant postdoc by the name of Taylor Steeves. So Taylor and I worked together on all kinds of stuff. And since Wetmore was working on ferns, why I had to go ahead and work on ferns. Well, that was all right. So I chose this one, Osmunda cinnamomea, the cinnamon fern. Really beautiful. All of the fertile fronds are completely separate from the sterile. It's a fairly primitive fern. And the thing I tackled was the mechanism of leaf unrolling. So this is what it looks like over a period of about 3 weeks. And if I had been Paul Green, I would have gotten all kinds of mathematical analysis. But as it was, I decided to first to describe the phenomenon, where it was cell division, where it was cell elongation, the role of auxin, and where the auxin was coming from. And it all worked out pretty well. So I got a thesis out of it. And in the process, one of my advisors was Kenneth Tiemann, who was a giant in the plant hormone field.